Okay, morning guys. Where is everyone? Could be the rain.
The construction.
The construction.
It's really bad.
Yeah, I know. I know. That's why I got delayed as well. All right, we'll get started. And today we're going to be talking about positive sense RNAs. We saw, for example, coronavirus last week, and this week we are going to look at these three set of viruses: picornavirus, flavivirus, and toga viruses. Okay? So before we get to the lecture, don't forget your midterm is next week, Wednesday, 5 to 7 p.m. in HL 170. So it's going to cover lectures 1 to 5. So it does not cover today's lecture. Okay? So by the time you guys are done your quiz this week, you should have a review for all 5 lectures. Okay? All right. So midterm will have multiple choice, fill in the blanks, and short answer questions like I told you guys before. And one thing I would say is also understand the material. Don't just memorize stuff. Okay? Because short answers usually are mostly application. You'll have some definition and fill in the blanks, but it's going to be mostly application questions. Okay? And a couple of things that I don't release about the exam is how many questions are on the exam. It doesn't matter. You have 2 hours to do the exam, you'll have more than enough time. Okay? And I'm not going to say how many short answers versus multiple choice either. Majority of it will be multiple choice, but you will have quite a bit of short answers as well. Okay? All right. Any questions before we start? Yeah. Study guide is your lectures. I don't have a separate study guide. Okay? You guys can go through your quizzes again to see how you answer the questions. Okay? Speaking of which, the quiz questions would be roughly about average difficulty you'll see on the exam. Some questions will be easier, obviously, and some questions will be more difficult. Okay? But you can accept, or you can expect roughly about the same average difficulty. Okay? All right. Any other questions before we start? Okay. All right, so we'll start with picornaviruses, and these are very small RNA viruses. Okay, so that's why they're called pico or picornaviruses, and these are non-enveloped viruses. What that means is it's naked icosahedral capsid. So that means the capsid itself is how the virus works. It does not have an envelope. Very small diameter, anywhere roughly about 30 nanometers, could be slightly more or less. But there are hundreds of these species, viral species, that infect humans, mammals, birds, etc. Okay? And what you'll notice is they cause fairly common diseases. So things like hepatitis, myocarditis, cold, common cold, polio in humans, okay? So what we're going to do is we are going to mostly focus on how poliovirus works, but picornaviruses, they tend to have similar or identical replication cycles. So you have linear positive sense single-stranded RNA genome, and because the virus itself is fairly small, the genome size is small as well, so roughly about 7 to about 9 kilobases, okay? It has a single open reading frame that encodes a polyprotein. That polyprotein then gets processed, and from there on, you can start packaging the virus. So here's a EM image of what poliovirus looks like. This is a 3D model based on the data. So this is based on the actual protein data. And this is what the viral structure will look like. So again, you can see how it's organized. So you have your viral proteins 1, 2, and 3, and how they get organized within this capsid. Now here is the genome. Yeah? A 3D model is basically using studies like X-ray crystallography and cryo-EM, stuff like that. Okay? So here's the genome. Notice something different in the genome here? Anything? Yeah? Well, there is a protein. There's a protein right here on the 5' end of the genome. It's called the VPG, okay? And it does have a poly A tail like you guys have seen before. So here's the little poly A tail, okay? Now, these picorna viruses, they bind to cellular receptors using their depressions and loop regions on the surface. So let's go back for a second. So you see how some stuff is sticking out, and then you also have depressions in the structure of this virus. So this is what will bind to the cellular receptors, okay? So these depressions or loop regions on the surface, they play a key role in binding to the cellular receptors, okay? We saw some examples in one of the earlier lectures where receptor, for example, gets inside that binding pocket in the canyon of the virus. So same concept here, okay? Now the mature virion will have these 4 proteins, VP1 to VP4. Could be 3 to 4 proteins depending on the virus, okay? Now the key thing here is that the VPG protein is bound to the 5' end of the RNA. This RNA does not have a 5' cap, so as a result, VPG is attached covalently to that 5' end, okay? And it has a very short poly A tail at the 3' end, okay? So what function do you think VPG will serve in the absence of the cap? Because here's your VPG, here's your non-coding region on the 5' end, and it has a specific site called IRES, or internal ribosome entry site. And we're gonna go into the details of this in a bit. Yeah? Okay, so ribosome, yes, partly, but also it's the protection of the mRNA from the 5' end as well, okay? So then you have your capsid proteins, your VP1, 2, 3, 4. You have a small leader peptide. Then you have your various other proteins. So you have your non-structural proteins, as well as, lastly, you have your non-coding region on the 3' end. So both on 5' end and 3' end, you have non-coding regions, okay? The 3' end is slightly smaller than the 5' end, but what you'll notice is the poly A tail. In this case, normally the enzyme that adds poly A to mRNA in the cell is poly A polymerase, okay? But in this case, the poly A tail is actually encoded by the genome. So there is a sequence in the genome of the virus itself that once it's replicated, it adds that poly A tail into the RNA, okay? So we'll look through these proteins and we'll see how this works. All right, so your VP1 to 3, they will form the capsid shell, and VP4, it actually remains buried within that shell, okay? So it'll be towards the inside of the shell as well, okay? Then you translate these genes as a single polypeptide, okay? So what that means is you're gonna synthesize one polypeptide, and then you cleave that polypeptide to form those functional peptides. So you undergo proteolytic cleavage, and there are 1 to 3 proteinases that will do that, and there are 6 to 8 replication proteins that are synthesized by this genome, okay? So for example, the P1 region that's here, that encodes the structural protein. So proteins that will make up the capsid, okay? P2 and P3 encode non-structural proteins. And you'll see here is that VPG is encoded by the genome. So once it's encoded by the genome, it will bind to the viral RNA, as you can see here, okay? With me so far? Okay. All right, so first thing I'm gonna show you guys is this internal ribosome entry site, okay? So the 5' non-coding region of the genome has this IRS site, which allows the initiation of translation without having a cap, okay? So your ribosomes need to look at the cap, and then they will start translating that RNA. But in this case, we don't have a cap. So we have an internal ribosome entry site, which you can see in this figure right here, okay? So it will allow for translation at somewhat of an internal site, so it's just a little bit inwards on the 5' end, downstream of the 5' end, and it has extensive secondary and tertiary structures. What does that mean? That it has extensive structures, secondary and tertiary structures, yeah?
Because of maybe the bonding, it kind of assumes a 3D shape, kind of like with tRNA.
Okay, so we have the genomic molecule in that region base pairing, okay? So you form these hydrogen bonds and form these secondary and tertiary structures like we see in tRNA, okay? These structures can interact with a number of cellular proteins to aid in the translation of this RNA. Once you make your polyprotein, your viral proteases will cleave them into either precursor molecules or mature proteins. So let's have a look. So you have your genomic sequence here. You make one polyprotein. And then you're going to make all these different types of cleavages to synthesize various other viral proteins, okay? So don't worry about the details yet because we're gonna go into that, but understand that you're making that polyprotein once the ribosome enters in this IRS signal, okay? And let's look at an example. So just like I said, there are extensive secondary and tertiary structures in these IRS elements. And there are multiple types of IRS sequences. And in this figure, you can see type 1 and 2 sequences. So type 1, again, not to worry too much about the detail. What you have is various regions along with your start codon. Okay, so here's your start codon. And all these IRS elements, they have a pyrimidine-rich tract, roughly about 20 to 25 nucleotides away from the start codon, okay? And this likely the function is to initiate the translation at the right spot. So basically, this IRS sequence along with these pyrimidine-rich tracts, you can see that here. Oops. All right, you can see that here. You can see the A-rich loop here, and here's your pyrimidine-rich tracts. So you can see this is extensive secondary structure, but here's another start codon, the real AUG. Okay, so you're gonna make the polyprotein. You have this pyrimidine-rich tract, and this will basically help position your ribosome at the correct spot for translation, okay? Now, these virions, they bind to cellular receptors using canyons or loop regions on the surface like we discussed. And your VP1 to 4, they all form the jelly roll structure. So they form 8-stranded beta barrels, so this is the beta jelly roll structure like we discussed in one of the earlier lectures, okay? Now, the genome itself, it can pass through the pores that are formed in the cell membrane, either in the cell membrane by capsid proteins, or they are internalized by receptor-mediated endocytosis. So they can get in the endosome as well. So either they inject their genome directly from the cell membrane, or they undergo receptor-mediated endocytosis. And the capsid will dissociate after the endosome acidifies, and eventually the genome will go out, okay? So just to show you guys an example, here are the different viruses, picorna viruses, and what they bind to in the cell. So you can see that there's a variety of different receptors that these viruses can bind to. And you can look at some of the characteristics. See anything common? What's common in the characteristics? The immunoglobulin superfamily, okay? So they belong to the immunoglobulin family of proteins. A lot of these receptors, they belong to that family, okay? But not to say that the other types of receptor don't exist, they do. In this example, what you're looking at is mostly immunoglobulin superfamily, okay? So how does this work? Well, once your poliovirus binds, the RNA goes into the cytoplasm after a major rearrangement of the viral structure. So let's look at the figure. Here's your genomic RNA. This is your VPG bound to genomic RNA, okay? So now pay attention to what happens to VP4. So here's your VP1 and the dimer of VP1, and this is your VP4. This little bar here is your VP4, okay? Now, as the endosome, for example, acidifies or you find the right receptor, what's going to happen is this protein is going to insert itself into the membrane, okay? So see how the conformation change happened and how that conformation change pushed part of the protein into the target membrane. In this case, it's the cell membrane, okay? Once you do that, you're gonna form a pore, and as you form the pore, you're gonna send this RNA out into the cytosol, okay? What would I call this particular part of the protein? Let's see if you remember your lectures. What's the part that attaches to the membrane or inserts itself into the membrane? Yeah. The fusion peptide or the fusogen, okay? So that's your fusion peptide in this case. Only thing is, this does not have an envelope, so it's not really going to fuse the membranes of the virus and your cell membrane. So this kind of conformation change, again, is going to create a pore in the membrane, and then it will send the RNA through the cytosol. Now, the problem is, most of the cellular RNAs, they are translated in a cap-dependent mechanism because there are set of proteins that bind to the 5' cap of the mRNA, okay? So, for example, here's your cap, and then you have your initiation factors. So the various translation initiation factors. So in this case, you have your EIF4A, 4E, 4G, okay? EIF basically means eukaryotic initiation factor, okay? Now, they form this complex called the 4F, and it binds to the 5' end, and then it recruits the ribosome, the small subunit of the ribosome. So you have all this complex bound to the 5' end of the RNA, and this is called the pre-initiation complex, okay? Now, once you do this, your 40S subunit will move along. It will scan for the start codon, which is your AUG, as you can see here. AUG encodes for methionine, okay? So what does that tell you about proteins? All proteins that are synthesized in the cell, yeah? They all start with methionine, okay? So that's not just the viral proteins, it's also the cellular proteins as well. Once it finds the start codon, your 60S subunit is going to bind and forms the full ribosomal initiation complex. So that's the ATS initiation complex. And then you can start translation. This is cellular RNA, not the viral RNA, okay? Now, question for you guys is, what's the S value? Why do we say it's 40S or 60S and then it forms 80S? Sedimentation coefficient, okay? So that's how these components sediment when you're trying to carry out centrifugation under density gradient, okay? So basically this gives you roughly the size of the complex, and now the translation can begin and the cell will go and translate the mRNA. But when it comes to the virus, problem is we don't have a cap. So how do we assemble this complex? Well, once the cell is infected by picornavirus, it causes a proteolytic cleavage of your EF or IF4G complex, okay? So the virus itself is going to cause cleavage of this whole complex, okay? As a result, it's going to abolish the cap-dependent translation in the host cell. So what that means is, it's hijacking the cellular translation. As a result, cell cannot translate its own mRNA, okay? Your 4E may bind to other components, it may be sequestered, so such that it becomes unavailable to start translation. So that sequestration of 4E, it will aid in stopping this process, so as a result, translation won't start. Your host proteins will bind to IRS, and then it will help with the docking of the 40S subunit. So what you have done now is you have interfered with this complex. As a result, it won't go to try and translate the cellular mRNAs. But your various host cell factors will recognize this RS sequence, and it will recruit the 40S subunit of the ribosome as well. Again, your 40S scans the RNA for initiator codon, so you start codon. Then you get your large subunit coming in, you assemble the full ribosome, and you can start carrying out translation, okay? So basically what this virus is doing is hijacking the cellular translation process. So the cell basically stops translating its own mRNA and starts translating the viral mRNA after infection, okay? Now, remember that we talked about the proteins, and they are made as a single precursor polyprotein, and these are cleaved autocatalytically by viral proteinases. So some of these proteinases, they will cleave themselves to become active, okay? A lot of these viral proteases or proteinases, they will become active and they start cleaving polyprotein, okay? First, you cleave it into precursor proteins, P1, P2, and P3. P1 then gets cleaved into capsid proteins, and from there on, P2 and P3 get cleaved into non-structured proteins, such as proteases, okay? One thing you'll notice here is the RDRP, which is the RNA-dependent RNA polymerase. Why do we need this? Why do we need RNA-dependent RNA polymerase? Yeah?
Because the cell it's needed to replicate in the cell that only has DNA-dependent RNA polymerase.
Okay, you need to replicate the genome, okay? Cell does not have RNA-dependent RNA polymerase. Cells have DNA-dependent RNA polymerases, okay? So this is part of the viral genome as well, so you're going to encode it to replicate your RNA, okay? Now let's have a look and see how this works. So after you synthesize your viral proteins, your viral RNA gets replicated. Now, a couple of things here. Notice here that it's attached to a membrane vesicle, okay? And you have your VPG and your various other proteins bound to the positive strand genomic RNA, okay? Now, most proteins that you make from P2 and P3, they are involved in replication of the genome. And the way it works is you initiate the replication using this multi-protein complex that is bound to cellular vesicles. Where else have you seen this? So here's your membrane vesicle, and you have a complex that binds and holds the RNA to those vesicles. Yeah, we saw it in coronaviruses, for example, okay? But that does happen in a lot of other viruses as well, okay? So since these vesicles are required for replication, once you have an infection with these viruses, these membrane structures start to reorganize, and there's a proliferation of vesicles once the infection takes hold. As a result, it works as a nucleation site for viral replication. So these become your viral factories once they are initiated, okay? So first thing you're going to do is, since it's a positive strand genome, you're going to make a negative strand complementary copy of that RNA. This is called the antigenome, okay? So you make the antigenome and use that antigenome as a template to synthesize positive strand RNA, okay? So here's your positive strand RNA, and look at what's happening here. You have your poly A tail, and then you're going to replicate that into complementary sequences. So since it's poly A, your negative genome, your antigenome will have a poly U on the 5' end. Now, VPG is also attached to this 5' end, which is you can see right here as well. And once that happens, your polymerase will start to make a replicative copy. So what you get is a replicative intermediate. So this is protein 3D, okay? Now this RNA synthesis is primed by the VPG that's covalently bound to uridine residues in your anti-genome, okay? So this allows it to hybridize to the poly A tail and serves as a primer for the negative strand synthesis, okay? So this VPG is attached to these U's. But notice here that you have a couple of U's here that are complementary to that A or those A's. And this is where the VPG will get attached. So you attach it to uridine, it's called uridylation, okay? And as you continue synthesis, you're gonna make a negative antigenomic RNA, okay? So you start reading the template on 3' end, synthesize, and once you have this negative strand antigenomic RNA, now you can start making copies of that RNA, okay? So as a result, what you're going to make is the positive strand progeny genomes. So let's have a look here. So you have multiple polymerases copying the genome. And on this end, you have the 2 A's. Obviously, you're going to make 2 U's for that. So on this end of the genome, on the 5' end of the actual positive strand genome, you have these 2 U's that get added, okay? And these basically will result in formation of this kind of structure, okay? Now again, your VPG needs to be attached, okay? So you attach the VPG to this UU, And see what's happening here. You see these two U's? That's what's base pairing with your positive strand genome, okay? So as a result, you're gonna make multiple copies, and now you have replicated genome, okay? Now, one thing I would ask you guys here is, which one do you think is predominant in the cell? The positive strand or the negative strand genome? Positive? Yes, why? Okay, very good. So your negative strand is being used as a template, and as a result, you're gonna make multiple copies of the positive strand. So the negative strand genome or the antigenome roughly is about 1 to 2% of the viral RNAs that you'll find in the cell. Mostly it's positive strand. Okay, with me so far? All right. All right, so now you have synthesized your proteins, processed them into the right kind of proteins, and then you replicated the genome. So now you have your genome and your protein, so that means you can start assembling the virus, okay? So there's a precursor protein called VP0, which gets processed to VP2 and VP4. So here's your VP0, VP3, and VP1 here. Now, this needs to be cleaved, and this is cleaved by a specific proteinase. It's called a 3C or 3CD proteinase. So again, if you go back to the genome and look at where these proteins are, you should be able to identify which of these fragments encode for a proteinase. Now, your various viral proteins, they assemble into what's called protomers, okay? Protomers are basically subunits that are composed of different polypeptide chains. So you cleave this proteolytically, and then you form this, for example, a 5S protomer, which then assembles into a pentamer. And from there on, the structure becomes more and more complex, okay? So your 5 of these protomers will assemble into a 14S pentamer. Pentamers, as well as the RNA, they assemble into what's called the provirion, okay? So this is a provirion. What that means is it's not a fully mature virus yet, okay? Now, how does the RNA get into the virion? Well, there are possibly different pathways that do this. One is you thread the RNA genome into procapsid. How would we do that? You've seen that example before. How does that work? You have a procapsid, which is empty, and you're gonna send this RNA in, thread that into that procapsid. How does that work? Who remembers that figure? Anyone? Yeah.
Is it through a concatemer, and then there's a terminase that cuts it?
Okay, so in that specific case, you had concatemers, and there was a terminase that basically pushes the genome inside the capsid, cuts it at the right sequences. In this case, that's not really the case. You basically make this RNA genome, and then you thread that just in a similar manner, okay? So you'll have the capsid, and you'll basically fill in that capsid with that RNA. That's one possible mechanism. The other one is that the pentamer associates with the genomic RNA. So it's actually attached to the genomic RNA. And once it's there, it's going to assemble into a provirion, okay? Again, remember, this is a provirion. What that means is it needs to be further processed to actually make the mature virion. So how does that processing take place? Basically, you cleave the VP0 to VP2 and VP4. So this is the last step in maturation. where you're cleaving the VP0 protein into VP2 and VP4, okay? And as a result, what you get is the mature virion, okay? Everyone clear? All right. So here's your progeny RNA getting in, your pentamer forming the procapsid, and this is the second mechanism where you can thread that in. Okay, so either one of these can work. And if you look at the infection, what we see is that the infection with picornaviruses, it inhibits several of the host cell macromolecular functions. They shut off the host cap-dependent mechanism. How do they do that? Let's just see if you remember what we just talked about. How does it stop the cap-dependent mechanism? Okay, so it cleaves the pre-initiation complex, which has proteins like EIF4, etc. Okay? It induces formation of these cytoplasmic vesicles, like we saw earlier, and it alters intracellular transport pathways. And by altering the pathways, the virus can move around in the cell as well. And once that's done, newly synthesized virions are released from cell by lysis. And at this point, they're ready to go infect other cells. So here are the different steps: attachment, then entry of the genome, translation, making the polyproteins, forming those vesicles, and then replicating the genome, okay? So this has all the steps of the virion's life cycle, okay? So if you guys wanna look at the different steps, they're all listed here, okay? So you guys can read through that, and you should be able to explain this whole process, okay? So you now know how picornaviruses work. So let's go over the steps. What's step one? Look at the figure. Binding. Then what? Okay, you send the genome in, or you induce receptor-mediated endocytosis, and you are in the endosome. Then what? What happens then? Yeah. Sorry, say that again. Okay, you're gonna make proteins, you're gonna translate proteins, the viral proteins, and then what? Okay, to translate the viral proteins, you need to inhibit your cellular mRNA translation. So then what? Yeah. Replicate the genome. But how do we replicate the genome? We first need to make antigenome. Okay? Then you make the genome, then you assemble the virion, and then it can lyse the cell and exit out. Okay? Everyone clear on that? Okay. So that completes picornaviruses. Next thing we're gonna look at is flaviviruses. The name comes from flavus in Latin, which means yellow, and it was called flaviviruses because it can cause things like jaundice from yellow fever infections, okay? It's a spherical enveloped particle, slightly larger than picornaviruses, so in this case it's about 50 nanometers. The nucleocapsid inside is smaller, it's about 25 to 30 nanometers, has an icosahedral symmetry, and it's spherical, okay? Now, one thing to note here is that the surface is relatively smooth, okay? So this is an image of the dengue virus, which is a type of flavivirus, and this is the model structure. There are no projections from this surface, from the envelope. Instead, it has a golf ball-like appearance. Your enveloped glycoproteins, they are arranged such that they are in icosahedral symmetry. So here's your M protein, your C protein, you have a dimer of E proteins, and here's your genomic RNA. But look at how the glycoproteins are arranged. Rather than as spikes, in this case, they are arranged in, again, icosahedral symmetry, and that gives the virus its smoother surface, okay? The T number is 3, okay? Everyone clear? All right, so T number in this case is not for capsid. This is for the envelope proteins and how they are organized. That should tell you how many subunits you need, okay? All right, so flaviviruses, they are generally transmitted by arthropods, okay? So they are also called arboviruses, and they can cause several important human diseases. These viruses infect humans, they can infect animals like monkeys and birds. You also have pestivirus genus, and this is mostly relevant in livestock. So cattle, sheep, etc. They don't have insect vectors, and humans are not really infected. Then we have the Hepacivirus genus that has a single virus, the hepatitis C virus, okay? Again, no insect vectors, but it can go from person to person through things like blood transfusion, IV needles, sexual contact, so through body fluids, okay? And here are the different genuses and the name of the different viruses, the host, and the kind of diseases they can cause. So as you can see, they do cause fairly intense diseases. So for example, hemorrhagic fever, encephalitis, swelling of the brain, okay, etc. So they are very important, and they do cause several important human diseases, okay? So if you look at the structure of the genome or the virus itself, it has an envelope, and your envelope proteins are arranged with icosahedral symmetry. You have 180 copies of your M and E proteins. So remember the T number was 3. That means you have 180 copies of your envelope matrix and envelope protein. So you have your envelope protein heterodimers. When the virus is immature, it can actually show spikes on its surface, as you can see here. But as the virus matures, it becomes smoother and smoother. So it has a golf ball-like appearance, okay? So again, this is the morphology of the dengue virus. And again, at neutral pH, you have proteins that are arranged perpendicular to the surface, so they are sticking out. But as it transits through Golgi network, etc., the pH decreases, and what you get is this kind of structure, okay? Now, this kind of structure doesn't fully form yet. You do need to cleave it, and that peptide is called the PR peptide, as we will see, okay? So you get to this structure, then you cleave a protein, and it becomes golf ball-like, okay? All right, let's look at the genome. It's, again, a positive sense linear single-stranded RNA genome. A bit larger, about 10 to 11 kilobases. It has a cap on the 5' end, but the feature that's different here is that it does not have a poly A tail, okay? So there's no poly A tail at the 3' end, as you can see here, okay? Again, you translate all genes as a polyprotein, and then you cleave the polyprotein to form capsid protein, envelope proteins, and non-structural proteins. So there are 3 structural proteins and 7 non-structural proteins, okay? So again, another thing you'll notice is here's a cap, here's the 5' region, untranslated region, and here's the 3' untranslated region. But notice that there is no poly A tail. And again, these structural proteins, all these sites that you see, this is where different proteases and proteinases will cleave the polyprotein, okay? Now, if you look at how the genome is organized, it's very similar to what we see for picornaviruses. But this is very different from togaviruses, as we'll see later, okay? The virion morphology is different, genome sizes or morphology is similar, genome size is similar, and they do get transmitted by arthropods, okay? So mosquitoes, for example. Now, again, your RNA is translated into a single long polyprotein. It needs to get processed, just like we have seen before, so proteolytic cleavage. And you require both your viral and cellular proteinases. So here's your 3' end, secondary structures on the 3' end, here's 5' end, various proteins, and that's how it gets synthesized. See how it's still attached to the membrane as it's being synthesized? And all these different arrows indicate where the protease cleavage will take place, okay? Everyone clear? Yes? Okay, let's take 10 minutes before I bombard you with more information.